So we want to welcome everyone today to our webinar. It's been a little while since we've been with you. My name is Beth Voss, and I am with the Choroideremia Research Foundation. And we just want to welcome you today. We want to thank our sponsor, which is 4DMT. Our placeholder did not come across, so I'll make sure I say a special thanks to 4D. Today we have some really incredible new research with new people, new faces, and I need to reflect my notes so I make sure I get the explanation right. So thank you everybody for letting me take a look at my notes. We're going to be learning today about new imaging research that hopefully is going to be able to be used as potential biomarkers for photoreceptor functions. Now, anybody can try to say that without looking at notes. I'd give you an A+. But anyway, today we have two great researchers. We have Dr. Niamh Wynne, who's with us from the the Medical College of Wisconsin in Milwaukee. Right, there we go. And we have Angela Zhu, who's the PhD candidate from the University of Pennsylvania, who works with a beloved person in our foundation, Dr. Jessica Morgan. So welcome, you guys. Wanted to say thank you. We have both ladies that are here today, and they're going to be showing their work and their PowerPoints. And I'll be, as always, monitoring the chat feature. And they both would like, if you have questions, just to go ahead and type them out using the chat or the Q&A, and I'll ask them right at the time. So since we have two different presentations, it's easier to ask at the time. So once again, welcome. And Niamh, I think you're going to start first, right?
Yep.
Okay, perfect. All right. Thanks, everybody.
Just check you can see everything correctly before I start. Okay. You can let me know if there's any questions or if something doesn't show up correctly. So firstly, thank you so much for the invitation to talk. I'm thrilled for the opportunity to share our research with you. I'll be talking specifically about retinal sensitivity in comparison to cone density in choroideremia. We thank our funding sources, Choroideremia Research Foundation, having funded some of the initial work that this research came out of. And I wanted to tell you a little bit about me to kind of give you some context as to where I'm coming from. So I did my medical degree at Trinity College in Dublin. In Ireland, I worked looking at genotype, phenotype correlations in all inherited retinal diseases across the island of Ireland. That research project was called Target 5000. And then I did some postdoctoral research at both here at the Medical College of Wisconsin and at the University of Pennsylvania, which is where the work that I'll talk with you today is from. I specialize particularly in adaptive optics, imaging, and its application in inherited retinal diseases. And so it's an area that I'm particularly passionate about and excited to share with you today. So this is an audience that I don't need to talk through all of the background of choroideremia, but it's helpful for us to take a look at the picture here and see the appearance of the retina at the back of the eye and the atrophy that we see, which is kind of death of the different layers of the retina, which gives us this pale appearance. And the optic nerve is here with the squiggly lines are the vessels, arteries, and veins. And so in choroideremia, we see death of photoreceptors, retinal pigment epithelium, and choriocapillaris, which are three of the layers of the retina. But we get central spared vision, which is here, as we can see. And this sparing of a small island in the center is part of what I'll talk about in the next few slides. So when it comes to clinical trials and treatments for inherited retinal diseases, we would like to have something to use to monitor whether a treatment has worked that changes over the time course of a trial, which is typically 2 to 3 years, has the potential to improve and is clinically relevant and meaningful and makes a difference to people's quality of life. And part of the problem in choroideremia is if we have a small area where there's spared central vision that we don't expect to change over that 2 to 3 year period, we can't show an improvement or even prevention of further progression within this small area. which poses a challenge, not only in choroideremia, but in inherited retinal diseases. So because of this preserved central vision, the visual acuity, or the dark letters on a white background that we test at clinic visits, isn't a useful measure for tracking treatment response, which leads us to using structural measures, measuring how much retina is here. And this requires a better understanding of the relationship between the structure and the function. And just to kind of delineate what I mean by structure and function, because sometimes these words can get thrown around and they don't mean much. Structure is like whether or not there's a light bulb there. And then the function is when you flip the switch, whether or not the light bulb turns on. And so we apply that to retina, where we can see the retinal extent of different cell types and different imaging. And then we apply extra techniques to see whether or not those cell types are functioning or working, or how well they are functioning. And that's something that we can track over time, and might be something where we can capture some information and see if there's been some turnaround or some improvement in the function. Which leads me to adaptive optics, which is a advanced retinal imaging type. And it was first adapted from telescopes looking at the stars, where the Earth's atmosphere forms a curved surface. And the telescopes had advanced technology that could compensate for the changes and aberrations in that curve to be able to see things out in the space with better definition. And then it was turned towards the eye, where we could compensate for aberrations in the curve of the cornea to allow us to see individual photoreceptors. We can see individual cones on a very small scale that previously would have only been visible using histology. This technology has been around for a while, but we've been improving it and understanding it, and it's a very exciting advance. So looking at the retina in individuals who have choroideremia and what the difference is from individuals who have healthy retinas, here's a typical image that we get after an hour and a half imaging session with a person from one eye in a person with choroideremia. We see a central area. This is the fovea, or the macula, the part that's responsible for central vision, and then arms spreading out. And after we've taken the images, we stitch these together almost like a jigsaw puzzle. You can see the jagged edges here. When we zoom in on this particular area, which is away from the macula is where the cones are most densely packed, and away from the macula, they get further and further out, and the cones get wider and bigger themselves. So as we look in here, we can see these circular structures, which are individual photoreceptors in the eye. Out here, there's an absence of those circular structures, where we can see that there's an atrophic area. So that's an area where the photoreceptors have died, and we can no longer see those circles. So using adaptive optics, we have learned a lot with previous studies about the appearance of the retina in choroideremia and how it works. So we know that photoreceptors are absent in the atrophic retina, or the part that doesn't work or people can't see. The photoreceptors are reduced in number within the retained functioning retina, and that individual photoreceptor morphology is altered in choroideremia. And we also know that the photoreceptor mosaic varies qualitatively from healthy controls. So even where the numbers of the cells are the same, the appearance of the cells is different. Microperimetry is more of the function test. So where we talked about the light bulb and whether or not it's there, the function test is the light bulb working, or are the cells working? And microperimetry is a test that, unlike adaptive optics, can be performed in the clinic and is regularly performed in the clinic. It's commercially available. There are several different commercially available devices that can test microperimetry. It usually involves showing a light of different intensity and different regions, and deliberately showing that light to specific areas within the retina. So it's a clinically available tool, and it can assess the sensitivity at discrete regions, like I've talked about. And we can see plotted here on a retinal picture from an individual with choroideremia, the points where the retinal sensitivity has been measured. And we can see within the area of retained retina, there are some higher responses represented in green, yellow, orange, and that the red responses are recorded out in these areas where there's atrophy or the cells have died. So when it came to analyzing the two different types of information that we have from the retina as part of this study, we aligned the adaptive optics images to the microperimetry responses so that we could take very, very small regions of interest from the retina and examine the count of how many photoreceptors, or those circular structures, there were in the same region where the responses were recorded from. So we have a computer program that helps us to identify these, but we go back through and manually check them to see that each circle is truly represents a photoreceptor. And then we generate an estimate of the cone density. So that is how many cones we can see within a particular area. So we can see here, we had 31 eyes from 31 patients included in the study. We had 314 individual regions of interest across 6 different eccentricities. And that really means where they are in relation to the center of the macula. And we know that the photoreceptor density decreases as we get away from the center of the macula. And so the eccentricity was important to know. And we measure them out to 2 millimeters, which is a very small distance from the center of the retina, but there's a very large change in the number and the appearance of the cells, as you can see here on the right, from the center all the way out to 2 millimeters or 2000 microns. The age range of the participants that we had in the study was age 10 to 57 years of age. And the median or the mean visual acuity was 80 ETDRS letters, which is easier and more understandable as 20/25 in Snellen acuity, with a range of 20/16, which is better than 20/20 vision, to 2100, which is relatively poor central vision. And like I said, we can see the difference in how many cells there are. These regions are all the same size on the retina. And at the center of the fovea, the cones are much smaller, where we extend out to the far from the fovea, or the center of the macula, the cells become bigger. So when we compared the cone density in choroideremia to healthy control retinas, we could see that the cone density was reduced across all eccentricities. So this tells us that there are photoreceptors being lost throughout the retained area in the center of the retina, and not only at the atrophic edge. And the reduction was actually greatest in the central region of fixation, because this is where healthy controls have the highest number of cones, or the highest density of cones. So there's about a 50% reduction here in the center. We also looked at the visual sensitivity as measured on that microperimetry response, where we were showing lights and asking when people could see them as we increase the intensity of the light. And across all regions, so both in the center, the superior, inferior, and lateral sectors, there was a reduction in the sensitivity in choroideremia as compared to healthy controls, and this was statistically significant. So that comes to the question that we really posed with this study, which is, are structure and function related? Because if we want to follow structure in a clinical trial, we need to know how that relates to whether or not people have vision and how good their vision is. And we showed that sensitivity and density are in fact correlated at fixation, and this is statistically significant. So we can see that the y-axis here represents sensitivity, with 20 being the highest sensitivity, or being able to see the lowest brightness of light. And then the cone density, which means the greater number of light bulbs or photoreceptor cells, if you like, along the x-axis. And we can see as the number of cells increased, so too did the sensitivity. This was just at fixation. So we looked across all of the points from fixation to 2 millimeters, as I showed earlier. And we see the same relationship exists, although it's more scattered when we get to 400 microns from the fovea and 800 microns from the fovea. It's also worth seeing that this x-axis goes out to 100 here at the fixation point, but it's shorter for the other points. So there's a smaller spread of densities as we spread out from fixation, which is something that we see in healthy control retinas, but also in choroideremia. It's just worth noting as we go through the rest of the plots. And we see that further out from the center of the retina, the range of densities gets even smaller, which again is something that we see in healthy controls. But we also see that the relationship between the density and the function or sensitivity is lost. So we don't have a statistically significant relationship between them as the numbers get lower. When we looked at the overall picture, so this is a montage or a jigsaw puzzle of images that we've taken of the photoreceptor mosaic from one individual participant with choroideremia. We can see that the center of the macula would be here on the right of this panel, and we extend out to the atrophic area on the left. And here the cells near to the center have the highest sensitivity. So they're on the green scale, meaning that they could see the lowest intensity lights here, and they extend out to where right at the border, they couldn't see even the brightest light. So we noticed that as we looked across all of the participants and all of the 314 areas that we had examined, that there was this trend of decreasing sensitivity as you went further from the center, which we noted also meant that they would be getting closer to that atrophic border, because the atrophic border comes from the outside in. And so the further away from the macula you get, the closer you get to that atrophic border or zone of dead cells. So we looked at the data again, and we examined as we are further from the atrophic border, that the sensitivity is better. And as we approach the atrophic border, the sensitivity reduces. And we could see that at fixation, that wasn't true. And again, that's the area where we had the highest number of cells across everybody. We have a significant outlier here, but there's not the same relationship as we can see as we extend out of the fovea. This measurement at 2 millimeters has a p-value of 0.06, meaning that it's trending towards statistical significance, but it is not, in fact, significant, which may be due to the fact that we have the fewest number of regions of interest examined at this distance from the fovea, meaning that fewer of our participants had remaining retina at this distance out from the fovea. So perhaps with more, we would find that to be statistically significant. So in summary, we see that retinal sensitivity increases with cone density and increasing distance from the atrophic border. And the cells in the perifovea can be structurally present with reduced sensitivity. So therapeutic strategies aimed at preserving cone structure in the fovea may prevent progression. And functional outcome measures, so that whether or not the light bulb is working, it's best employed in the perifovea and not in the center where we didn't see that relationship. I'd like to thank the members of the Morgan Lab where I conducted this research, and I'm happy to take any questions.
We don't have any right now, actually, unless somebody wants to chime in right now and type their question in. That was really good. Thank you so much. That was a great explanation. Just super.
Thank you.
Angela's clapping.
Should I start mine if
I think
no...
you know what, here, there's one question that just came in. Oh, it says, very interesting and easy to understand. Thank you, Niamh.
Yay.
Thank you.
Perfect, thank you. Yeah, Angela, why don't you go ahead?
Sure, I'll start sharing my slides.
We can see it.
Cool. And my talk is it's in some way kind of build up on what Niamh has already talked about. So I might skim through some of the same background knowledge that Niamh already had a really clear and easy explanation on. So, but if anything's unclear, please feel free to type in the question or have Beth interrupt me at any point. So without further ado, I'm gonna start my portion of the talk, which also talks about the retinal structure and function in choroideremia using the adaptive optic scanning light ophthalmoscope that you just talked about. And a little bit about myself. My name is Perelman. I also go by Angela. I am a fifth-year PhD candidate at Dr. Jessica Morgan's lab at University of Pennsylvania, which maybe some of the patient families are pretty familiar with, because we get a lot of your support by coming in and being a wonderful research participant at our institution. So before I start, I would like to just show you the disclosures, the financial disclosures that our lab and the people involved in this research had. And I'd like to say a special thank you to the Dubra lab at Stanford, who helped us with all the instruments. And then Dr. Tomas Alleman, who is a great clinical retinal specialist at UPenn, who helped with all of the clinical side of the work, and everybody else in the Morgan lab at Penn. And here are all the funding sources that supported this research. And without further ado, I'm going to go and talk about the technology that we use and the exciting research that happened on choroideremia. So as Niamh just talked about, the instrument we use is called adaptive optic scanning light ophthalmoscope. And she talked about all the basics and how the science behind it. So I'm not going to dig into the details, but here I have a montage, or the jigsaw puzzle that we've just talked about, of a normal-sighted patient, normal-sighted participant. From the left here is the macular region, and or what we call a fovea, which we have it's a small region where we have the highest density of photoreceptor cells in the retina. And then going to the right is going out away from the fovea region in this person's retina. And we can appreciate, using the confocal modality of this instrument, we can get these reflective cone cells showing up on our image. So every single bright dot here inside this image is a cone photoreceptor cell. And on the left here, you can see the densely packed appearance of these photoreceptors. And as you move out away from the fovea, you'll see the density start to decrease a little bit. And then as you move further out, you can see the cone seems bigger. So every one of these big dots is a cone photoreceptor. And there are tiny dots surrounding it. They are called rod photoreceptors. And as you go further and further out, you can see the cones are a little bigger, and then they're more sparsely packed compared to the fovea. And the technology that I'll be talking about to examine the photoreceptor function is called optoretinography. So optoretinography is a technique for measuring the optical signal that arises from the fovea in response to a visible stimulus. So how we acquire it is that we image the person's retina with a near infrared light, and then we flash a very bright green light as a visible stimulus to stimulate the photoreceptor cells. And then as we flash the green light, which corresponds to this little bar here, that's the duration of that green light. And we can see that before we flash the green light, the cells are kind of just fluctuating, doing their own thing. But once we flash the green light, the cells either brightens or darkens or show some fluctuation in their brightness, which is the signal that we pick up and try to measure as a indication of photoreceptor function. And I'd like to add that this technique is actually it's kind of similar to an ERG, which maybe a lot of you haven't experienced with. So it just measures the signal that comes from the cells once you're stimulated, but this is non-invasive. So we do not poke anything into your eye. It's just literally looking through a microscope through your eye. And it only requires a couple seconds of video recording instead of taking a long time. So we're hoping this can be a very good technique for examining photoreceptor cells in people. And I don't think this audience needs a lot of background knowledge of choroideremia, but on the right here, I just have two fundus photos of two different people. On the top here, we have a normal-sighted person's fovea, the fundus photo, and on the bottom here we have a CHM person's fundus photo. And again, because we see these atrophic border encroaching in as in the CHM people and the region of interest. So the area we're interested in is the central remaining area where the photoreceptors are still functioning and intact. And here are two example images of the photoreceptor cells in a normal-sighted person on the left and a CHM person on the right here. So very immediately we can see there's a difference in the appearance of the photoreceptor mosaic. So on the left here in the normal person, you can see the cones are very evenly distributed with a pretty good density, whereas on the right here in the CHM person, we can see patches of cones that are extra bright and patches of cones that are kind of just dimmer than the average. So that's where we start to wonder. So if they seem to be there, but they have a different appearance, do they function differently as well in CHM compared to normal-sided control? And so these are the questions we're going to try to answer using optoretinography in choroideremia. So the 3 questions I'm going to be answering in this talk, first of all, is the optoretinography, abbreviated as ORG in our field of experts. I don't know why they picked, I think, or some people think ORG is funny. Apparently I found out this conference, I don't know why. So ORG in CHM versus normal-sided participants, are they different? And secondly, is ORG correlated with structural phenotype in CHM? And lastly, does ORG correlate with clinical measurements of retinal sensitivity, which Niamh just briefly talked about? So before I talk about the details of results and answer these questions, I would like to just say a little bit of how we set up this experiment. We had 4 normal-sided participants and 10 CHM participants participating in this research. We performed AOSLO imaging, so we did the structure imaging that Niamh just talked about and made the montage, and we performed the ORG measurement in what we call parafovea, which is around 150 micron to like 300 micrometers away from the fovea. It's very close to the fovea. And the stimulus we used was a very bright green light for a duration of 2 seconds during the video recording. So here I have a video of a raw video that we collected in one of the normal-sided participants. I hope the video plays through the Zoom. So here we can see that's what we see on the computer when we're collecting the videos, and you can appreciate there are some distortions from the scanning nature of the instrument, and there will be some movements from the eye because we can't hold them 100% steady. So before we do any analysis, we need to perform some post-image processing to remove the distortion that's coming from the scanning and the movement. So after we do all these post-processing, a result of the video looks like this. So after the processing, we can make sure the cones stay in the same place throughout the frames and throughout every single video we collect, so we can pick out the signal from the cones easily. And now I'm just gonna zoom into this little squared region to show a little more detail of the cones. So here is that region zoomed in a little bit so we can see the nice photoreceptors on this image. And to extract the ORG signal, I have to first identify where the cones are. So I use the same software that Niamh just talked about to pick out the center of these cones. And then I manually adjust for any of the misidentified cones. And to extract the mathematical signal out of it, so I calculate the mean pixel intensity of a 3x3 pixel region around each of the blue dots, so the cone centers that I identified. And I perform a normalization because there will be fluctuations in the image. Also, I divide the cone reflectance value by the mean of all the cones in that frame. So we eliminate the effect of movements or dimming and brightening of the image throughout the video. And then lastly, I standardize the cone signal. So I standardize the signal to its pre-stimulus mean. So as a result, before the stimulus, the signal will be relatively flat and considered as like the control pre-stimulus no signal. And then we can see a nice increase of the signal, which I'll show you later. So here's a video I made to help you visualize the signal. So before the stimulus, every cone is just fluctuating happily. And then once the stimulus is presented, you can see the cones either brightens or darkens. I'm just gonna play it again. They fluctuate, and then the stimulus comes on, they brighten and darkens. And to make that into a mathematical plot, I just plotted out. So every trace here, every single thin line here corresponds to the reflectance signal on each dot here on this video. And to extract a aggregated measurement out of this messy traces, I calculate the standard deviation at every single time point in this trace. And then I subtract the control signal. So we also collected videos without any stimulus. So just normal, no green flash. And we subtract that signal out of the stimulated videos. And as a result, I get a nice curve here. And what we call this a ORG response from the visible stimulus. And then later on, if I talk about the amplitude of this response, it will be the peak of this curve. So now finally we can answer these questions. First of all, how different is optoretinography in CHM versus normal-sighted participants? On the left here, I have the normal-sighted participants, and on the right here, I have all the CHM participant signals. And we can clearly see that there is a significant decrease in the height that the curve can go to, and the time is also slightly different. So there's a sharp increase, whereas here we do not see that sharp increase. And if I extract the peak value, which is what we call the amplitude of the signal, and compare it using statistical tests, we can find out that indeed CHM ORG signals are significantly lower in the parafovea compared to normal-sighted controls. And secondly, is this signal correlated with structural phenotypes in CHM? So Niamh just talked about a lot of measures we can make with the structural phenotype in CHM. And I would like to see if the response I'm getting from the ORG experiments have any correlation with the structure measurements. So first of all, I would like to show you the 10 CHM participants parafovea image. So on the left to right, top to bottom, I ranked everybody by their amplitude of ORG signal. And I'd like to zoom in onto the 3 that has the highest ORG signal. Although you may not be very familiar with these images, but based on what I've shown you earlier, you can see that all of these 3 people had a fovea cone photoreceptor mosaic that kind of resembles a normal healthy retina with a pretty much even distribution of the cones and a very densely packed appearance. Whereas if I zoom into 2 participants with the lowest ORG signal, we can see a more typical CHM fovea cone mosaic appearance with the really bright patches and the dark patches of the photoreceptors. So to quantify these phenotypes, I tried to extract the cone density measurements, so the number of cones in the same area, which Niamh talked about that she made. So when we compare the density and ORG, we saw a positive correlation with between the 2 measurements, and the correlation coefficient was 0.69. And I just like to bring up the normal-sighted image and the CHM image again, just to appreciate the difference in the density and the cone appearance. So if we plot normal-sighted participants' cone density versus ORG on the same graph, it will be all the way up here. And we can see there are some people have a density that is very close to a normal healthy participant. However, their ORG is still very low on this graph, meaning even if their density is similar to healthy normal, they still have a decreased ORG signal. And to make sure that our signal is not reduced because we are including less cones in the analysis, I randomly sampled half of the cones in the normal-sighted participants, so we kind of simulate a situation where they also have reduced number of cones like CHM participants. However, we did not find a significant difference in the ORG signal. So we know that the ORG signal is truly decreased because of the response of the cell remeasured is lower, not because of the number of cells included in the analysis is lower. And then, so that was looking at the photoreceptors from like an en face view, looking straight into the eye. Now we're wondering what about the structure of cones from a side view? So a cone looks like a little stick, or like they are not a flat 2D shape. They are a 3D structure in your eye. So usually we can measure what we call the outer segment length of the cone. I'm not going to go into too much detail, but you're welcome to Google it later. So we believe that the ORG signal is arise from the change in the morphological shape of that outer segment, the length of the outer segment. So whenever it's stimulated, the length changes, and that's causing the optical path to change for the light that travels in and out of your retina, which causes the reflectance, so the brightness change of the cone when we're looking at it en face. So to look into that, we pull out every CHM patients and the normal-sighted participants' OCT image. These are acquired just using a clinical OCT device. So it doesn't have the optimal resolution to actually resolve every single cone's outer segment. But so in this case, we try to get a proxy measurement of the outer segment length. So here are two example OCTs from the healthy normal control eye and a CHM eye. And we can appreciate some features like there's a layer called interdigitation zone. It's slightly disrupted in the CHM. So we don't see the clear lamination in CHM. So to compromise, we choose to use a measurement called ellipsoid zone to RPE, so retinal pigment epithelium that Niamh talked about, and / Brooks membrane complex, which is a bright peak just here as a proxy measure of the outer segment length. So I would just zoom into this normal-sighted participant's OCT image. So how we took the measurement is that we find out the peak of this OCT image that corresponds to the Brooks membrane or the RPE layer, and we find the peak that corresponds to the ellipsoid zone, which is where the outer segment starts. And we subtract the distance between these, we get the distance between these two peaks. And here I have an example of that normal image versus the CHM image. And we can see that the peaks are closer together in CHM, so the distance is a little bit lower, which might be indicative of the fact that the outer segment in CHM might be shorter, but it is a proxy measure, so we don't have a definite conclusion from it. And so when we compare this distance measurement to the ORG signals I just talked about earlier, we can see that, again, this distance is positively correlated to ORG signal, meaning that if we have a higher distance, then we have a higher ORG signal. And the correlation coefficient is 0.72. So it's a medium to strong correlation between the two measurements. And again, if we plot the normal-sighted controls there, they're going to be all the way up here. So they have a thicker, larger distance, so thicker or outer segment, and their ORG are also much higher. So the conclusions for structures, we can see that in CHM patients, the reduced ORG amplitudes correlated with the cone densities and the easy to RPE BRM distance. And lastly, how does ORG correlate with clinical measurements of retinal sensitivity? Thanks to Niamh, everybody here should be pretty familiar with the microperimetry measurements. So the green, darker green means that you see better. You can see the dimmer lights, you can resolve dimmer lights, and the lighter green or red or yellow just means a lower sensitivity. And so we tried to take out the foveal sensitivity measurement, which is the closest measurement to where we measure the ORG, and try to see if there's a correlation between this measurement and our ORG measurement. And indeed, again, we observed that ORG is positively correlated with foveal sensitivity measured using a clinical device called NIDAC MP1. Maybe some of you are pretty familiar with it. And the correlation coefficient is, again, a medium strong correlation at 0.73. In conclusion, ORG amplitude is significantly reduced in CHM paraphobia compared to normal-sighted controls. And the reduced ORG amplitudes in CHM correlated with cone densities and easy to RPE BRM distances in CHM, even though even in the patients with near normal density and distance. And lastly, ORG amplitude correlated with foveal sensitivity in CHM measured with clinical devices, showing its promise to use to assess retinal function. And with that, I would like to conclude my talk and take any questions that you may have. Thank you.
Well done. Well done in a challenging background, Angela. That was awesome.
Thank you.
Okay. All right, here we go. Here's a couple questions. It looks like, Niamh, of course the photoreceptors are dying, but why are the photoreceptors bigger in the periphery on CHM patients? Do the few remaining photoreceptors get bloated, or is it because they're less densely packed? And do they spread out?
This is a great question, and it's something that we've talked about in the lab. I know we've had these discussions with Angela based on her paper, where she showed that the roundness of the cells were different between healthy controls and choroideremia subjects or participants. So it's not, there's no definitive answer, and we haven't been able to study that. We would likely like to image somebody from very young to an older age with choroideremia with this device in order to be able to get a definitive answer. I think, Angela, didn't you do some calculations on if we had a normal density?
So the roundness of the cell versus density?
Yeah, but and didn't you do some hypothetical calculations of what size the cells would have been if we had a closer to normal density when you were working on the discussion for that paper?
Yeah, I think I did that, and our conclusion for the size of the cells out farther away is that we believe there are two possibilities in our discussion for that. One is that they might be showing signs of unhealthiness or dying, that they might, because a lot of cells might swell a little bit before when they're under stress, or when they're going apoptosis, which is the cell death process. And the another explanation is that because in histology, in biology in general, when you put cells on a place, they would like to take as much as possible. So they will expand to fill any space that's available around them. And that's what we believe is a very possible explanation also, that because the cells are reduced in CHM, especially in the outside area, they try to expand and take up all the space. And that's the
basically
two
the two hypotheses that this anonymous attendee proposed are correct, and that's kind of our thinking as well. We haven't designed an experiment to be able to definitively point to one versus the other as the reason for the cells changing in size, but those are two of our top thoughts.
Okay, here's a couple more questions. Are you able to show differences in sensitivity over time to prove or disprove the efficacy of potential therapies? Good question.
This is a good question.
Yeah, it is a very good question. And that's exactly why we're trying to develop this technique. These 10 patients I looked at, none of even if some of them went through therapy, I looked at the non-treated eyes. So these are all just natural, non-treated process. But we believe this technology will have the potential to assess the smaller changes in visual function compared to the other outcome measures. Because I know a lot of therapies only compares the visual acuity, but all 10 of the subjects, they either have 20/20 vision or 20/18, really good visual acuity. But then but the changing visual acuity doesn't really reflect the tiny changes that the therapy might be targeting, because the therapies are all cellular level. We're targeting single cells. It just can't be reflected from the visual acuity.
I think as well, it's interesting, even should the ORG, that work has some time to fill in, but the microperimetry responses that we have now, if you look at the areas where you couldn't see robust responses, despite the fact that the number of cells were the same as in other participants where there were good responses, it tells us that the structure is there and maybe there's room for improvement of the function, which would be something that could be captured in response to a treatment. And that's what, when the FDA looks to approve treatments, they want to see that there's been improvement in discrete pre-selected areas. And if those pre-selected areas are from places that we don't expect there to be any change because the sensitivity is already very high, then a trial is doomed not to show any results from the beginning. Whereas if we know that there's an area where there is structure, so there are cells that can improve that are not functioning to their maximum, then potentially we could select those regions at the beginning of a trial to follow over time. And there may be the room for improvement because you've got the cells that can improve. And we know that that number of cells can give us a good response as in other participants. So it's kind of that dissociation of the structure and function means there's light bulbs there that maybe we could fix or restore the function of, as opposed to dead cells where we can't, unfortunately, with gene therapies help.
That is a fabulous explanation. And I think that piece alone would be great to show people. And as we have viewers, that's great. That was a great question and great answer, guys. Thanks. Again, another question from the previous writer. Angela, if a CHM therapy is successful, that's what we're hoping for, would you be expecting the cones to rise up to a normal level or just to the higher level of a CHM patient? That is a big difference between all the normal versus CHMers.
This is a very good question. Yeah, we don't have an answer to that question yet because we haven't really looked into the ORG measurements from therapy patients that went through therapy. But I would say like a dream result for the therapy would to bring the cones response to the normal level, but we just don't have a definite answer of whether or not it's achievable. But any improvement I would think would be great. So
I think it's also worth noting that the responses, and you've said this already, but I'm going to reiterate it because I think it's important, is that the vision for those participants was normal. So even though their responses were much lower, they're still maintaining a normal vision with black letters on a white background. We all know that that's not real life, but they could maintain some pretty good function on that visual acuity measurement. And so maybe what's normal is more than what's necessary to provide good vision. And so even if we could demonstrate an improvement, it might maintain people's vision in a realm where they're happy and comfortable and have good quality of life, even if we don't get them to be perfectly normal on these tests.
Good answer.
Thanks, Niamh. That's a great point.
All right, I'm going to give everybody one more minute before we close our webinar for the day for any more questions.
Questions are fantastic.
Yeah, they were great.
Angela, until we, in this 1 minute, why don't you tell us where you're at?
Oh, I am in Argentina right now, Buenos Aires. I'm at a vision research conference called ICER, International Society for Eye Research. So my background's blurred right now because if I don't blur it, you'll see bottles of wines because I found a cafe in the lobby of the hotel to sit in to give this webinar.
Oh, it's wonderful that you joined us. We just thank you so much that you took time to do this.
No, thank you. I appreciate this opportunity. I'd like to meet all the CHM patients and families, and of course you guys too at the foundation. So thank you.
Oh. The person that asked, we had a couple people, but the same individual is asking a personal question. How did the two of you decide to pursue retinal research for your careers? Do you have family members with vision loss? Your enthusiasm for your work is palpable. I would agree. It is great to see such energy from our young and female researchers. Hear, hear. Thank you very much. So how about you answer that question and we'll shut it down for the day.
Niamh, do you want to go first or?
Sure. I guess I found my way into ophthalmology because the eye is so aesthetically beautiful and there's so much that we can do for people with ophthalmology. And particularly then inherited retinal disease research. I just met the most wonderful people through doing that research, and I met whole families because they would have been involved in our genotyping research. And I felt that there was a real sense of being lost in the winds of when there were no treatments even on the horizon. Patients had been discharged from clinics and had lost faith in their eye care providers or in the care that they were receiving, like the care as opposed to the medicines that they were treated with. And I really felt that there was a niche or a space for me because I had encountered this and treated people and conversed with people and understood what was going on. And I just care. And I think that there's so much difference that can be made with all the emerging treatments. And then once I found adaptive optics and saw its application in inherited retinal disease, I was hooked. But yeah, it's good to see that the passion comes across, as I hope to be around inherited disease and this research for my career.
Great.
So for me, I've always found the eye really, really cool. And I, as a kid, because I have very high myopia, so very nearsighted, which is very common in Asian, I think. So one of my family friend is a ophthalmologist, and she carries a lot of myopia research. And as a kid, I would go to her her facility, her hospital, to do all kinds of tests for her research projects, and I've always found that really cool. And then when I moved to the Americas, when I moved to the States, I stayed with this one host family, and the father of that family, he has macular degeneration. And so I saw him throughout the years. He used to be able to like use his peripheral vision to still read some books, and then he started to use the magnifying glasses to read the books, and then eventually he had to turn to audiobooks in total. So I was like, oh, this is so sad. I wish he can just read his book normally. So I naturally just went into what, like, my interest for eye research just grew. And then luckily in undergrad, a cornea researcher like approached me, and she's like, you have such great imaging techniques, so why don't you help me image some mouse cornea? I was like, okay, that's really cool. So I did some mouse eye research, and then I encountered Jessica Morgan's lab at UPenn when I was doing online research for graduate study, and I was also again hooked to the adaptive optics technique, and that's why I'm here and how I end up here. And I hope to do more retina research in the future.
That's just so great. Well, to the both of you, we just so appreciate it. And as we sometimes say, we're just so thankful and grateful that you're on our team, on the bench with us, working to help us find a treatment, and or to find the things that you're doing to help the endpoints that other researchers will be able to use. That's very significant. So just awesome. Thanks, guys. Thanks so much. And we hope to check in with you again in the future. Have a wonderful time in Argentina. Yeah, everybody says it's just chiming in. Thank you to you both. And we just are grateful. Thanks so much, everybody. And we're doing another webinar next week. So hopefully we'll see you then. All right.
Thanks,
Bye. Thank you so much.
Thank you.
Bye.
Bye.